Do you ever wonder about the mysteries hidden within our DNA? What could be the purpose of those billions of other letters of DNA that seem to have no function? They were once termed junk DNA, but is that really the case? In the disco-fueled decade of the 1970s, biologists first peered into the vast expanse of human genes. Amidst this genetic landscape, small pieces of DNA, known as exons, that coded for proteins, seemed like tiny islands in an ocean of genetic gibberish. The rest of the DNA, billions upon billions of letters, appeared to serve no apparent purpose. This seemingly useless DNA was unceremoniously dubbed junk DNA, a term that has lingered in the realm of human genetics for decades. Fast forward to the new millennium, when the first rough draft of the human genome was presented. The results seemed to affirm the notion of junk DNA, with a staggering 97% of our DNA appearing to have no obvious function. But is there more than meets the eye? To shed more light on this fascinating subject, we have with us Ewan Birney, a leading computational biologist. Welcome, Ewan. Let's delve right into it. Our earlier views of the genome organization, were they too simplistic? Ewan Birney, renowned computational biologist and the head of the European Bioinformatics Institute, responds, "People always knew there was more there than protein coding genes. It was always clear that there was regulation. What we didn't know was just quite how extensive this was." Birney leads us into the fascinating world of the ENCODE project, an endeavor that has turned our understanding of DNA on its head. The project has discovered that the so-called junk DNA is far from being useless. Instead, it's teeming with crucial genetic elements. About 1.2% of the bases are in protein-coding exons, explains Birney. People speculated that maybe there's the same amount again involved in regulation, or maybe a little bit more. But even if we take quite a conservative view from our ENCODE data, we end up with something like 8 to 9% of the bases of the genome involved in doing something like regulation. This revelation is nothing short of revolutionary. It implies that much more of the genome is devoted to regulating genes than to the protein-coding genes themselves. And that 9% can't be the whole story, Birney continues. The most aggressive view of the amount we've sampled is 50%, so certainly it's going to go above 9%, and one could easily argue for something like 20%. That's not an unfeasible number. The term junk DNA now appears to be a misnomer, a relic of our past ignorance. Birney asserts, "Yes, I really think this phrase does need to be totally expunged from the lexicon. It was a slightly throwaway phrase to describe very interesting phenomena that were discovered in the 1970s. I am now convinced that it's just not a very useful way of describing what's going on." So it seems the term junk DNA is a misnomer, but what does this imply? Continuing with our discussion, Ewan, should we retire the phrase junk DNA now? Indeed, Ewan Birney, leading computational biologist, believes it's time to put the term junk DNA to rest. During the 70s, the term was used to describe the seemingly non-functional parts of our DNA, but today, as Birney asserts, this descriptor is far from accurate and fails to capture the complexity and potential of these genomic regions. Birney elaborates, "The phrase junk DNA was a slightly throwaway term to describe very interesting phenomena discovered back in the 70s, but I am now convinced that it's just not a very useful way of describing what's going on." So what is going on? Well, it turns out that these junk portions of our DNA are not junk at all. In fact, they play a critical role in regulating genes. To give you an idea of the scope, while only about 1.2% of the bases in our DNA are in protein-coding exons, Birney suggests that around 8 to 9% are involved in regulation, a conservative estimate based on the ENCODE project data. Birney further explains that 9% can't be the whole story. The most aggressive view of the amount we've sampled is 50%, so certainly it's going to go above 9%, and one could easily argue for something like 20%. That's not an unfeasible number. In other words, a significant portion of our DNA is devoted to regulating genes, a function far from the junk label initially assigned. This revelation not only redefines our understanding of the human genome, but also opens up a new world of possibilities for understanding human inheritance and disease. The term junk DNA definitely seems to be outdated, Thank you for your insights, Ewan. As we delve deeper into the complexities of our DNA, it's clear that every bit of our genetic code has the potential to be a hidden treasure, waiting to be discovered. It's clear that what we once considered junk DNA holds hidden treasures. This understanding has been redefined by the incredible work of Ewan Birney and the ENCODE project team. As we've learned, our DNA is far from being a heavily padded text. Instead, it's a complex, intricate tapestry woven with genetic codes, switches, and signals. The landscape of our genes is not filled with genetic gibberish as previously thought. Rather, it's teeming with important genetic elements, once dismissed due to our limited understanding. The concept of junk DNA was born in the 1970s, a term coined to describe the seemingly useless parts of our genetic makeup. But today, we know better. The ENCODE project has shown us that this so-called junk is far from worthless. Out of the 3.2 billion bases in human DNA, only 1.2% are in protein-coding exons. Yet their research indicates that around 8 to 9% of the genome is involved in something like regulation. This figure may even rise to 20%, completely overhauling the way we view our DNA. The vast majority of our genetic code, once dismissed as junk, is now understood to be rich in regulatory elements, switches, and signals, each playing a crucial role in our genetic makeup. These hidden treasures in our DNA are involved in regulating our genes, a function as critical as the protein-coding genes themselves. This groundbreaking insight undeniably underscores the importance of further research in this field. The more we delve into the mysteries of our DNA, the more we realize just how much we have yet to understand. It's a reminder of the infinite possibilities that lie within our genetic code, waiting to be discovered. So, it's time we retire the term junk DNA. It's not a useful descriptor for the complex and intricate genetic elements that make up our DNA. In the vast sea of our DNA, nothing is truly junk. Every piece has a story to tell, a secret to reveal. It's a reminder that there's always more to learn, more to discover in the fascinating world of genetics.